And the next speaker is Krystyn van Vliet. Do I pronounce that right?
Van Vliet.
Van Vliet, right, pardon me. Who's the Paul Cook Career Development Associate Professor of Materials Science and Engineering at MIT. And Dr. Van Vliet focuses on the cell-material interface, as I understand it, and how local and mechanical states combine to alter biological processes.
Thank you. I do, but since this found it right away, let me bring up the presentation again. Okay. Fantastic. So I'm glad that we're continuing to do this, and I'm very happy to show you what we're working on. So I'll start with the acknowledgments here, just to point out that it's been great to collaborate with various folks at MIT, as well as Tufts Medical, Children's Hospital, and other hospitals in Singapore. But I'm very excited about this potential collaboration with Dana-Farber to move this forward. The resolution here is not matching up with my screen, so let me see if I can fix that quickly for you here. Great. So what we are interested in my group is actually looking at the coupling between chemistry and mechanics, and in particular at the cell-material interface. And that other material could be a drug, it could be another cell, it could be the extracellular matrix. But how the mechanics and chemistry go together to affect the cell function. And we do this through use of both experiments, typically tools that we build, and I'll tell you about some of those, as well as computation, because we're interested in mechanism. And in particular, we're interested in mechanisms related, oh, thank you, that's much better. In mechanisms related to cancer, including the process of angiogenesis. So what you're seeing here is an archetypal cartoon of the tumor that is surrounded by blood vessels that are growing towards it, and that's thought to be one of the critical mechanisms in cancer metastasis through angiogenesis. And there's quite a bit of complexity here in the interaction between ligands, such as, for example, the vascular endothelial growth factor, and receptors, such as those on the surface of the vascular endothelial cells that line the blood vessels. Okay? So that's one of many examples in this picture of reversible interactions between molecules that can include even the adhesive forces that hold the cells together and may affect, for example, leukocyte migration. There's also a lot of mechanical cues in this picture, few of which are depicted in actual biological cartoons. Okay, and that's what we and others are bringing to the picture here. This could include cell traction, and I'll show you an example of that. It could include the stiffness of the extracellular matrix, which is known and been shown to affect processes such as cell proliferation and differentiation. And fluid flow is yet another mechanical cue. So even though our focus is on those mechanical cues primarily, we also recognize the close coupling with the chemical cues. And this could include pH, and I'll show you an example of that, macromolecular crowding, as well as you would imagine, ligand concentration. Okay. So we're interested in how mechanics and chemistry together are coupled to affect, for example, binding kinetics and how those affect downstream functions that are relevant to cancer, primarily angiogenesis and metastasis. So I'll give you a few examples, starting with the molecular level and working our way up. One of the tools that we've made a lot of use of in the past several years and developed is an atomic force microscope, which has become a ubiquitous tool in characterization of materials as well as biological objects, if you're just taking pictures of the structure. But what we've done is move that forward by using the fact that the atomic force microscope is nothing but a cantilever that's made out of a material such as silicon, and added some chemical functionality to that. So what we do is we take a picture of the cell where we've decorated the end of this cantilever, which is about a hair's width long, okay, with individual molecules. And these could be drugs, they could be antibodies, they could be fluorophores. So whatever you're interested in interacting with the cell surface with, we chemically functionalize that on the end of this cantilevered probe. And then we take a picture that's based on mechanical force as well as binding to that particular ligand. And the measurements that we take at each pixel are the force as a function of the displacement away from the cell surface. So with this kind of tool, we've been able to do a few things that required also modeling. For example, we've been able to extract the kinetic binding rates, both the on rates and the off rates, of ligands that are bound to the probe to the cell surface. So we can see the receptors with molecular resolution, and that's shown here. Although the picture is small, I'll just say that the figure on the left is a picture of the cell using this functionalized probe, a portion of that cell. And this is a vascular endothelial cell, so we know it expresses receptors such as VEGF receptor, vascular endothelial growth factor receptor. So we put an antibody for that receptor onto the cantilever, and then each of the dark spots that you see, and I've just circled one in the center of this second picture, each of those dark spots represents the position of a receptor that's bound to the ligand on the cantilever. And by taking pictures in a time-lapse fashion, the same region over and over, while doing essentially a competitive inhibition experiment with the same antibody in solution, we can get the off rates and the on rates of those ligands, or antibodies in this case, to that receptor. Now, the implications of that have been very useful in working on problems in cancer, such as we recently worked on this to characterize an unknown at the time surface receptor for an anti-angiogenic peptide, loop 6. That was developed down the street at Children's Hospital by Marcia Moses, and we worked with her to help establish the mechanism of that drug by being able to show that that oligopeptide bound to a specific receptor. Okay, so this can be used to better understand the mechanisms and identify new targets. So I'll show you another example where we used the same tool and even took it further in understanding how other extracellular cues, such as the acidity of the extracellular environment, affect problems relevant to cancer. So if this is a cell, and it could be a cell within a primary tumor, or it could be a cell in the vasculature, right? The way that that cell adheres to the matrix is through transmembrane proteins called integrin receptors. So it's known that vascular endothelial cells, as well as particular engineered cells we showed, express an integrin known as alpha V beta 3, among others. It's also further known from the literature that the extracellular environment of a primary tumor is acidic, anywhere measurements as low as 5.4 to 7.0, right at the tumor periphery. Okay, so we asked the question whether or not this acidity would affect this binding between the cell and the matrix through those integrins. The first way we looked at that was with computational modeling. So this is an engineering tool, okay, but it's not for a physical experiment. In this case, we took the tripeptide of the matrix ligand, such as fibronectin, docked it to one of those integrins, and did a series of simulations called steered molecular dynamics for this binding interaction and unbinding interaction at normal physiological pH as well as acidic pH. And those simulations predicted that under acidic pH, this conformation went into the activated state, the open state, more often. So what this says was it predicted that the integrins would bind more readily, have an effective higher on rate in acidic pH than they would in physiological pH. And we use that same atomic force microscope approach, as I told you about earlier, to put that ligand, in this case RGD, we can also use fibronectin, on the cell surface. And you can see here that the frequency of binding, which is shown on the vertical axis, actually increases significantly at the acidic pH representative of the tumor. Okay? You can take that one step further and say, well, the tumor is not a uniformly acidic environment. In fact, you would expect that there's a gradient between the healthy tissue at the margin and the tumor itself. So we created pH gradients through a device like this. It's technically a microfluidic device, but there's, in this case, no flow. And this enables you to maintain a gradient of pH for long periods of time, at least over 24 hours, between a normal pH and an acidic pH. And through that, and again the use of modeling of the cell, we were able to show that the cells, in fact, polarize towards the acidic region, crawl towards the acidic region of the tumor. Okay? So this suggests to us, although there's many more things that should be done and will be done in terms of cancer cells, that it's not that primary cancer cells crawl towards the acid, but perhaps this is an attractor for the vascular endothelial cells to crawl towards the tumor. Okay. This model, I should mention, was originally developed by Laufenberger and co-workers. And although it looks like Joel's model, because it looks like it has receptors and capacitors in it, this is actually a mechanical model of the cell, so that you can actually predict migration. Those are springs and dashpots, not resistors and capacitors. So we found through this series of experiments from the molecular to the cellular level that vascular endothelial cells in the blood vessels actually exhibit faster integrin activation and therefore polarization towards acidic gradients. The last example that I'll show you is in another type of imaging that we do of cells under force, and I'll show you this for the example of parasites. So parasites, as you may know, are cells that go around, and that's where they get their name, go around blood vessels, particularly play a prominent role in the development and stabilization of capillaries. So here you're looking at a scanning electron micrograph of a capillary, and you see the parasites are on the outside, and the basement membrane is often between those parasites and the vascular endothelial cells beneath. That basement membrane is mechanically very interesting because if you pull on it, the stiffness changes, okay, and that's what this graph shows. So for example, if the cells pull a little, that basement membrane is not very stiff. If the cells or something else pulls a lot, that basement membrane becomes an order of magnitude more stiff. Okay? So we asked the question, would the parasites, which had recently been thought to be contractile cells, be able to pull on that matrix and maybe stiffen it? And we did that by again using the atomic force microscope, but here in a different way. So you see here an atomic force microscope image of a parasite while it's alive, so it's in body temperature, solution, controlled CO2. Right? So it's basically an AFM and an incubator. And these are the actin stress fibers that you see of the parasite. However, orthogonal to that, you see wrinkles. Okay? So we cultured these cells on a very thin membrane that had the same mechanical properties as the basement membrane. When the parasites contract, they wrinkle that membrane like you would wrinkle a carpet. Okay? And the atomic force microscope images those wrinkles. Because it's a 3D image, we can quantify the amplitude and wavelength of those wrinkles and then calculate the force the cells generate. Okay? So this led to two important conclusions for us. This work spread over a series of papers in collaboration with Ira Herman at Tufts, showed that parasites exhibit sufficient mechanical force to do at least two things that could promote angiogenesis. They directly strain the vascular endothelial cells to which they're attached, because that strain, that force they're exerting goes right through the basement membrane. And they also stiffen that basement membrane. So they change the mechanical environment of the cells. So we're looking into ways to aggregate that, understand it more, and see how it relates to the cancer microenvironment. So final slide here. In the limited time we have here, I just want to point out that we've been doing quite a bit of work in bone marrow and are very eager for collaborations in that area. For example, we've been working on rapid mechanical analysis of heterogeneous cell suspension, so the different cells that are within the bone marrow. And we've developed tools that can characterize various physical properties, although not the electrical ones, including size and stiffness. And the in vitro and in vivo results are very promising in terms of isolating specific cell types within the bone marrow. We've also characterized the bone marrow itself as a mechanical material, and we could do this on other tissue cell types of interest. And that has allowed us to actually figure out what the environment of the cell is and distinguish when it goes awry. So we look forward to any further discussions and collaborations in these areas. Thank you.
Questions for Dr. Van Vliet, please.
Yes, very nice talk. Does your AFM probe have enough spatial resolution that you could actually count receptors in the surface of the cell?
In fact, you can. So it does have that kind of spatial resolution. It has molecular resolution, in fact, submolecular resolution. So as a tool, it can, and you're limited really by the size of the molecule you put on the probe. So in the work that I showed you, we compare the number of receptors per cell measured by quantitative facts as compared to our measurement, and they agree quite well.
Does it take a long time to do one cell?
Yes, so if you did the whole cell, so that's a good point. So you're only doing a portion of the cell and extrapolating based on the unit surface area. The technique is not fast, so it's not a high throughput imaging technique. You would not image the whole cell this way.
Other questions? If not, thanks very much. Is there? Please. Could you speak up, please?
Is the polarization of the cells in a pH gradient analogous to the polarization of the cells in a gradient of matrix rigidity, for example, or is it the opposite?
So that's a very good observation, and we did argue in the paper it's very analogous to the durotaxis or chemotaxis. Yeah, so if the matrix were more adhesive, it would be analogous to that polarization as well. That's right.
Please.
The parasites in the tumor microenvironment differ antigenically from the parasites in other anatomic sites. So what you describe applies to parasites in different sites or this specific certain sites?
Right, so the comment is that parasites from different locations may behave very differently. So the data that I showed you is are parasites from the retina, and we're in the process of looking at parasites derived from other locations as well. So I don't have the answer to your question, but it's certainly of interest.
And if I may ask, you mentioned that the parasites can stimulate angiogenesis, but in the tumor microenvironment, you want to do just the opposite.
That's right. But once you
And so
understand the
how-
mechanism, you can go both ways. So we have ways to encourage the contraction of those parasites as well as knock it down. So yes, you're right. In wound healing versus cancer, you want two different outcomes, but we think we have ways to tweak those.
Okay, thanks very much.